In this lecture, we're going to talk about passive and active transport across the cell membrane. To review, equilibrium is when the concentration of a substance is equal throughout a space. So in this diagram, for example, you can see that by the time you get to the third beaker, you've reached equilibrium. It got there through diffusion, which is the movement of a substance from an area of high concentration to an area of lower concentration. So the ink in this example was highly concentrated when it was first dropped into the beaker, and then it spread out throughout the solution to places that were a lower concentration of ink. So remember, water will always move from an area of high concentration to an area of low concentration. Remember also the structure of the cell membrane. You have a phospholipid bilayer, so that's this structure here, and then it has within it proteins. So this, for example, is a protein that resides in the membrane. There's other proteins here and here that also are embedded in the cell membrane. Remember also that the cell membrane is selectively permeable, meaning it can choose what goes into and out of the cell. There are a few things that impact what can move across the cell membrane. So some of those things include size. So if the molecule is too large, it cannot cross the phospholipid bilayer, but perhaps it can go through a protein, or the charge of a molecule. If a molecule has a positive or negative charge, it has to go through a channel because it is polar, and the lipid bilayer lipids are nonpolar, and so those molecules cannot cross the lipid bilayer because they are repelled by the lipids from the phospholipids. So some small molecules require help from proteins. Large molecules do not cross except through a process we'll talk about later called exo or endocytosis. In order for things to get through a selective permeable membrane, they can either go about that by passive transport or active transport. Passive transport goes with the concentration gradient and does not require energy. Active transport goes against a concentration gradient and requires energy. So passive transport, again, no energy is required. It's when things move from an area of high concentration to low concentration, and they don't need energy for that to happen. That happens naturally. Examples include diffusion and osmosis, but there's also another way for things to get across the cell membrane. They can go through a protein through a process called facilitated diffusion. So it's diffusion through a protein that has a channel or something in it, but it still moves particles from high to low concentration, and so it does not require energy. It's facilitated because it uses a protein, however. This diagram shows an example of passive transport, and it's specifically facilitated diffusion, where things go from an area of high concentration to low concentration through a membrane protein. These molecules are too large to travel through the phospholipid bilayer, but they can flow through a protein to equalize the amount of solute outside and inside of the cell. There's a couple of different types of facilitated diffusion proteins. One of those is an ion channel, and it's a transport protein with a polar pore, so that's an opening that goes through the protein, and they can just travel through at their leisure. There's no energy required. Another type of facilitated diffusion protein is a carrier protein, and that protein changes shape when it moves things through. So it can open to one side of the cell membrane, and then you can see here a molecule then will move into the binding space. Then the protein changes shape and releases it on the other side. Now we'll talk about active transport. Again, this requires energy because it moves things against their concentration gradient. So they go from an area of low concentration to an area of high concentration. This happens through several different processes. It includes carrier proteins, endocytosis, and exocytosis. This diagram shows an example of active transport, and so these molecules are going to go through a protein. They're going to go against their concentration gradient. So they're going to start out with an area of low concentration and move to an area of high concentration. And it requires energy, that's what we have here, usually in the form of ATP, but it always moves things with energy against its concentration gradient. A specific example of this that you have in your body is the sodium-potassium pump. So it's a molecule that molecules first bind to the carrier protein. So in this example, you have your first solute, and it's going to bind to the protein in this binding site. And then you can see that ATP, the molecule, is going to come, and it's going to deliver a phosphate molecule, and then the rest is going to leave as ADP. The phosphate molecule stays attached to the protein, and it changes the protein shape. So now you can release the protein, or release the old molecule, and then it opens a different binding site. So you can see here, now we have a square-shaped binding site for the second solute to come. That will bind to the new binding site. The phosphate will release, and then it can release the protein changes shape, releases the second solute, and it opens once again the original binding site. And so this process is active because it requires ATP, which is an energy molecule. Another type of active transport is called endocytosis. This is when the cell surrounds and takes material in from its environment. So this diagram shows both endocytosis and exocytosis. We'll start with endocytosis. So endocytosis is over here on this side. You can see there's this little green molecule, and the cell wants to take that molecule inside to digest it, but it's too large to travel through the phospholipid bilayer. And so instead, when this molecule makes contact with the cell membrane wraps around it, and then it pinches off so that you get this structure here. This is called a vesicle. And vesicles house things that are inside of the cell but are still contained. Then to digest it, the vesicle can fuse with something called a lysosome and break it apart so that it can be used for energy or other purposes inside of the cell. The opposite of endocytosis is exocytosis. You can think of this as exiting the cell, and it's when cells expel or release large amounts of material. So in our diagram here, the other side, we have exocytosis occurring. So now we've got our vesicle here, and you can see that it contains digestion, or the products of digestion, and so it no longer needs those inside of the cell. So the vesicle is going to travel to the cell membrane, it's going to fuse with the cell membrane, and it's going to dump the contents outside of the cell where they're no longer needed. The last thing we need to talk about is cell communication. The proteins that are on or in cell membranes can have a variety of functions. Sometimes they act as receptors, so they can receive messages from the body or from the environment. They might also act as marker proteins that allow the cell to identify itself as part of your body or as foreign material like bacteria or something. First, we'll talk about receptor proteins. Receptor proteins are proteins that bind to specific signal molecules, and it allows the cell to respond to the signal molecule. So for example, this is an ion channel, and normally it's closed, but if a signal molecule binds to that receptor protein, it can change the permeability because now things can travel into the cell. If that signal molecule releases from this receptor protein, the ion channel will close again and it will no longer be permeable to those molecules. Receptor proteins can have a variety of functions. They can change the permeability. They can also act as second messengers. A second messenger is a part of the cell that releases a signal molecule inside of the cell. So the way that it works is that a receptor protein receives a signal molecule. So here you can see the signal molecule binding to the receptor protein. The signal molecule does not enter the cell, but the receptor protein, when it receives the signal, can tell an intermediate protein to tell an enzyme to do something. And then the enzyme can form something like a second messenger. And so second messengers are a way for a message to get into the cell without something crossing the membrane.